Good afternoon. It is afternoon, and I'm very pleased to be welcoming you to this week's Institute for Quantitative Biomedicine seminar. I'm Stephen Burley, the director of the Institute and the director of the RCSB Protein Data Bank. We are fortunate today to be hosting Iman Dehzangi from Rutgers Camden, and his title of his seminar could not be more appropriate. We've seen Nobel Prizes this week in physics for the statistical physics for the origins of machine learning, neural networks, etc. And then we saw this morning the announcement of the Nobel Prize in chemistry going to Baker and the two senior figures from Google DeepMind for advances in computational prediction of 3D protein structure from amino acid sequence alone, and a very, very exciting set of developments for structural biologists and in fact for all biologists. It may not have been apparent from the Nobel announcement this morning that the work that was done in the Baker lab, the work that was done at Google DeepMind could not have been done without the protein data bank. And this is freely acknowledged both by David and the DeepMind team. And I'm sure in the weeks to come that will be emphasized as people think more about the impact of the prizes. So a few housekeeping announcements first, please. Next slide. Just an advertisement for October the 16th. We're going to be hosting Ramon Miranda Quintana from the University of Florida. I hope that he and his family are not going to be too adversely affected by what's going on there right now. The double hit is going to be, I think, catastrophic. But planning ahead, and hoping for the best, we'll be hosting him on the 16th next week. Next slide, please. I want to embarrass and congratulate once again Erica McCarthy, who's in the audience, for the receipt of the 2024 IQB Inclusive Leadership Award in recognition of outstanding achievements in training and mentoring. Thank you, Erica. Let's move to the next slide, please. And there's an opportunity to either apply for or to nominate a colleague for the 2025 Inclusive Leadership Award. The nominations applications are due on the 31st of October. And if you have any questions or concerns, please don't hesitate to reach out to Michelle Sangera. Her email is provided at the bottom of the right panel of this slide. Next slide, please. A very brief shout out for the additional training activities that the PDB is hosting over the next 8 weeks or so. Please register if you're interested for the November 4th webinar that's being co-hosted by Sebastian Bittrich from UCSD and Dennis Peel from here at Rutgers, focusing on the use of binary CIF, which is a very compact means of working with PDB data and in fact being able to work across the entire archive. Next slide, please. And then on the 7th of October, Ezra Pasak is going to provide a virtual office hour principally for software developers to help them get ready for the advent of extended PDB IDs, which is coming soon to a theater near you. Next slide, please. As always, we're looking for talented scientific software developers, graduates, undergraduates to work at the RCSB Protein Data Bank. A number of jobs are open, including my job, to succeed me as director. That opening was re-advertised in Science towards the end of September. Please don't hesitate to reach out to colleagues who you think would be appropriate nominees and get their names in front of me. It's absolutely key that we have a smooth transition to an individual, she or he, who's going to take the PDB to even higher heights. Other opportunities are listed below the opening for my role. Next slide, please. As always, please be grateful and mindful of the contributions that are made by the IQB team depicted here. And if I can have the last slide, please. It's a pleasure for me to be able to host and introduce Professor Iman Dehzangi from Rutgers, Camden. Iman received BSc training in computer science in his native Iran in Shiraz, and then went on to master's in bioinformatics in Malaysia, at a very interesting locale, Cyberjaya. I guess that really means cyber as in cyber infrastructure. And then he was a PhD student in Griffith University in Brisbane, Australia. Another very, very nice locale on the Gold Coast of Australia, and then came to us as an assistant professor in the Department of Computer Science and member of the Center for Computational and Integrative Biology at Camden. You'll see from the work that he's going to talk about today that he's in the thick of it in terms of AI and machine learning, and I'm sure we're all going to learn an enormous amount about his contribution. So, machine learning solutions tailored to address diverse challenges in the fields of bioinformatics and computational biology. Your timing could not be better. Thank you for coming.
Thank you. Okay, great. Thank you very much. It's an honor to be here and present at this center. My name is Iman Dehzangi, Assistant Professor at the Department of Computer Science, Rutgers University, Camden. I'm also a member of Center for Computational and Integrative Biology. It's an honor to be here. I started my master in 2008. I was working in machine learning field, and my brother told me that there's a field called protein fold recognition. And by then, I started kind of like shifting towards an application of machine learning. And I heard about PDB, Protein Data Bank, and I started using it. And it's very interesting for me that 16 years later, I'm here at PDB Center, RCSB, presenting my work, which is the application of machine learning into computational biology. Just a few days after the Nobel Prize of Physics went to deep learning, and the other Nobel Prize went to protein structure prediction. Interesting story. So, my research is actually, in general, can be just like formulated as the application of machine learning, computer science, and computational biology with a focus on deep learning, feature extraction to tackle different challenging problems into biology, and not just biology, applications in different fields. So in this quad chart, as you see, my research has started with protein, local and global structure prediction, which I'm going to present some of the work that we did. Introduce Spider2, the model that we present for local structure prediction, and then move forward to talk about some of other researches that I'm doing, which are very happy to share with you, which are the cutting edge knowledge of deep learning and machine learning to tackle different problems. In this case, the arthropod types prediction using transfer and deep learning. Finally, working on kind of like presenting some of my other research on cancer research and genomics, in which we use, again, machine learning, this time unsupervised learning for cancer subtype identifications. So it is kind of like, it's a bit tricky to give an introduction on what is machine learning here, and then what is proteins. But bear with me. I'm going to prepare it for the later concept that I'm going to cover. So machine learning, you're all familiar with. There's different definition. My favorite is actually with, the second one with Kevin Murphy, which make it in a very simple way. The goal of machine learning is to develop a method that can automatically detect patterns in data, and then to use that uncovered pattern to predict future data or other outcome of interest. Reality, we try to formulate different tasks. Some of the tasks are hard to be formulated. We cannot find a formula. How we do that? We are just like using machine learning. What machine learning does, it actually uses a huge amount of data, or any data that is available based on the availability of the data, and try to just like use statistical model, mathematical model to find that formulation. sometimes, we cannot just like put it as a formula. It just formulated internally to find the patterns in the data, and then use that pattern from training data. It learned a model which actually trying to formulate the problem, and then we can use that pattern for future data, or in other words, learn based on past and present to use it to solve future problems. Bioinformatics, on the other hand, is generally can be defined as the application of any computational methods, such as statistics, mathematics, machine learning, computer science, to solve challenging problems in biology. Mine specifically, based on those that are specified, which is protein fold and structure class prediction problem. My early works during my master and PhD, and slightly during my postdoc. And then protein local and structure prediction, which is still ongoing, but like less active. But I moved to just like post-translational modification of the problems, identifying protein binding peptides, and shifting to just like protein interaction prediction. And other works related to just like activities directly focused on USDA, worked with US Department of Agriculture, actually cranberry production pipeline that we're trying to develop a model to predict, to separate rotten versus ripe cranberries, which is actually very pivotal study in the state of New Jersey. And using deep learning for other tasks, such as identifying fossils, arthropods from a standardized image. So a short background about proteins. Of course, it might be kind of like not that necessary here. That's why I kind of go a little bit faster. So proteins are considered the most important macromolecules that perform a wide range of vital roles in biological interactions. They consist of like mostly 20 amino acids, in which each has their central carbon, alpha carbon, their hydrogen bind amino group, carboxyl group, and finally a side chain that dictates its physical, chemical, and physicochemical functioning. Know that the proteins are a sequence of amino acids, but they don't function as a kind of like loose string, but they fold in complex 3D structure to be able to do their function. We call them native structure, 3D structure. As you see, all the references are from RCSP Protein Data Bank. And this one was a field that I started and becoming kind of like familiar with bioinformatics, and I started doing my research. I focused on protein fold and structural class prediction. As you know, protein tertiary structure can be predicted solely from its primary structure. To date, experimental, even now with using AlphaFold and all the computational models, experimental approach are the main sources to predict tertiary structure, to determine tertiary structure of the protein, such as X-ray crystallography or NMR. At this stage, thanks to all the sequencing advancement, we have over 248 million protein sequence in UniProt. While despite all the efforts that have been done all around the world, we have around 225,000 protein structures that are deposited in PDB or protein data bank. So there is a huge gap. There is still a demand for just like finding a fast, accurate computational methods to predict protein structure. Computational approach consists of homology or ab initio or trading approaches. But still, some other tasks are trying to just like, or tried, especially during the time that I started working on predicting protein structure indirectly, or better to say that to predict protein structure class or protein folds that were defined based on protein structure prediction subproblem. Protein fold and structure class prediction, which is on SCOP. The problem that we defined is that protein, by then, it was just like argued a lot that protein can be defined, can be categorized into less than 2,000 groups called fold. So being able to determine protein fold, it will enable us to understand its function relatively, or how it is related to other proteins. And this problem can be formulated in pattern recognition terminology. How? So we have a given input, we have a bunch of data that belong to multi-classes, like around 2,000 fold. So it can be defined as multi-class classification task that can be solved using attribute selection, finding the attributes that are most relevant or can provide more discriminatory information with respect to proteins. Then feature extraction, how to extract numerical values from those attributes. Finally, using classification that would be suitable to solve that problem. In the literature, by then, a wide range of machine learning approaches have been used to solve this problem, and a wide range of features were extracted to just like formulate proteins. So features generally extracted based on proteins can be categorized into 4 groups. Sequential, which are based on alphabetic sequence of the proteins, such as occurrence, meaning occurrence of each amino acid along the protein sequence. Physicochemical properties, which are extracted based on physical, chemical, or physicochemical properties of the amino acids, such as hydrophobicity, hydrophilicity, polarity, van der Waals volume, and many more. In fact, as you will see in one of my studies, I studied over 125 different physicochemical properties. Then structural properties, which are extracted based on the local structure of the proteins, such as secondary structure or torsion angles. Finally, evolutionary based features, which are extracted based on substitution score of the amino acids along the protein sequence with other amino acids, such as features extracted from BlueSum, PAM, or PSSM, or position specific scoring matrix. So during my PhD, my thesis or my hypothesis, which I'm still standing on, that is that due to biological similarities of these problems, protein fold and structural class, or even other problems like protein binding, protein interaction, if a model performs good in one of them, it should perform effective, if not very good, at least effective or satisfactory in other problems. In this way, the nature of those problems are still persist. And we can just like connect them together because they are connected. So therefore, we should be able to just like develop a model that work consistent on related problem. In this case, protein fold and a structural class. If a model works well on just one, it means that it's overtrained for one and it's not generalizable. So what I did, my hypothesis studied this one, a comprehensive study on physicochemical attributes of the amino acids, and then proposing novel classification technique. But novel classification, now there is a huge focus on deep learning approaches, which are great, but at the same time, there is another approach called ensemble classifier, which is still just like we consider now as a state of the art. So at every step, if we combine or amalgamate different learning models that perform well together, it is likely that we achieve better results. It's not just like amalgamate them together. There are like theory behind them, of course, but my idea was using ensemble classifier, which still, as you'll see, using that for my other studies too. And then proposing novel feature extraction method to capture local discriminatory information from all aspects of protein. So sequential, physicochemical, evolutionary, and structural. What I've noticed first is that usually tend to extract features from the whole protein sequence. What I was suggesting is that instead of just using the features from the whole protein sequence, it segments the protein sequence. And from each segment, it is more like mathematical base. So we segment the protein sequence, extract features from each segment, like 50% of the sequence, 10%, 5%, try different approaches in between. And then extraction based on the accumulative length, meaning that just not independent segments, but segments that carried over different segments. For example, first segment would be 5%, second segment would be 10%, which include the first 5% as well. So it just carried the correlation information as well. For the sake of time, I'm going to go a little bit faster in this part, but what I did, use segmentation-based approach to extract features from different segments based on different aspects, physicochemical properties, evolutionary, and structural class. I tried to extract different set of features, and finally, combining all together and trying to just build a model to be able to try on the problems that were enhanced, like protein fold and structure class. What I did, from the protein sequence, I used PSI BLAST and Spinix, a protein secondary structure predictor, to extract structural features without relying on protein structure. So from protein sequence, you predict the secondary structure, and we then extract features. So in this way, no information from the secondary structure was used, or just like a structure in total. I extracted PSSM matrix and a spinum and use this sequence and that matrix to extract different sort of features. So I kind of like transform the sequence based on those evolutionary and structural information. It is similar to something that we use for large language model or protein language models, which I'm going to refer later as well. And amalgamate those set of features, try different set of properties, including physicochemical properties, different set of features, and eventually came up with using support vector machine to predict protein fold. By the end of the project, I was able to just propose PSSM Spine S. Definitely not the most innovative name. I regret it, but I just wanted to just name it something. And with all those things combined together, I just extracted 388 features. Means based on each protein sequence, I used just 388 numerical values. Compared to the previous works, state of the art like ACC, PSSM-AC, as you see, they were not that much of innovative in naming either. But extracting 4,000 features, I was able to outperform their works on 3 different data sets, EDD, which consists of 800 proteins and belonging to 27 folds, TG, consisting of 1,500 proteins belonging to 30 different groups, and the last F110 that I extracted directly from SCOP by myself as well, consisting of 5,000 proteins belonging to 110 different folds. So in fact, it was a very nice simulation of a bigger problem. And I was able to outperform previous study on all 3, and for the first time, at least by then, reached to 80% prediction accuracy for a fold recognition problem with over 100 different, for a problem with 100 different folds. And then I kind of like validate my works on protein structural prediction, a structural class prediction problem, in which is defined as categorizing protein into just 4 different groups: all alpha, all beta, alpha beta, alpha plus beta, alpha multiplied beta. Again, since that's just a little bit of like older studies, I'm going a little bit faster. But since it is a foundation of what I'm doing now, just like thinking that it might be very interesting to present them. And you see the kind of like how the feature extraction evolved, how machine learning models are evolved. As you see, working on protein structure class, since it was a simpler problem, 4 classes, I was able to reach to 92.2% prediction accuracy. In fact, by far better than the previous results. These 2 are just like PSSMS and Spinus are also my models. So the results are just like significantly better than those studied using same set of features and same model was trained on a different problem. to actually validate that my model is generalizable. So meaning that if you'd be able to use nice modeling plus good validation, you're able to just like achieve satisfactory results on those two problems, and at the same time demonstrate that the consistency of the model. Since then, I was still being active on protein fold and structural class prediction problem. One of my not so recent but still active works is just like that we did in 2009 with my group. We proposed PyFits. It's a toolbar that you can use to extract features from the protein. So it's publicly available. You're most welcome to use. But we are still hoping to be able to just return back to protein modeling my group at Camden and CCIB. Later on, I was lucky enough to be able to work with Professor Yaoqi. Yaoqi Zhou was from Purdue University that just moved to Griffith University by then. And he was the one that was the lead of the group that proposed a spine X, the one that I use for protein local structure prediction. And since then, we shifted our focus to predict local structure of the protein instead of global structure. Why? Because that local structure is actually in many ways is much more important, as you will see later on, for peptide prediction, for post-translational modification, to see how proteins interact with other macromolecules. So protein tertiary structure predicts is strongly related to a protein, to its local backbone. So local backbone can be defined in different ways. One of them is secondary structure, in which is defined as categorizing local structure into three distinct groups, which are helical shape, strand shapes, and coil, or sometimes called as turns, in which are not structure connectors between strands and helix. But local structure can be presented in continuous manner as well, such as torsion angles, which we present them in different ways. For example, can define dihedral torsion angles as well. So dihedrals are phi and psi, the angles in between a central alpha carbon inside the protein with its surrounding macromolecules, or torsion angles like theta and tau, which are the angles between two or three neighboring amino acids. So internal and external in local structure prediction. Beside that, we can calculate accessible surface area, or number of contacts, meaning that the number of neighboring macromolecules to an alpha carbon, which are other representation of local structure, which I'm gonna talk about them in more detail later. To begin with, they proposed a deep learning model, so shifting from traditional machine learning models such as SVM to start using deep learning models to predict torsion angles, theta and tau. By then, to the best of our knowledge, no machine learning methods have been used for that prediction. What we did, we extracted features from PSSM and developed a simple deep learning model with 3 hidden layers and 150 nodes per layer, initialized with autostack encoder to predict torsion angles. Instead of directly predicting each angle, we predict their sine and cosine as a radius. And developing this model, in 2014, we were able to just like predict, we call that model a spider, which relatively predicted theta and tau accurate compared to its previous models or predecessors. As you see, the native theta and native tau compared to what we predict were kind of like very close. But it was just like the pioneer, the first stage to predict theta and tau. Later on, we moved forward and extended our work to predict not just torsion angles, but several local structure simultaneously, predicting accessible surface area, secondary structure, torsion angles. Again, with that hypothesis that if a model performs well on predicting secondary structure, if it wants to be a good representative of predicting local structure, it should be able to do good on other aspect of the local structure. So for this aspect, again, we use physicochemical properties and position-specific scoring matrix, giving that to a deep learning model that was trained iteratively by several times, in which it converged after the third iteration to predict all those values. And eventually, we are able to outperform all the competitors, Spider1, Scorpion, Spinix, and PSI-PRED, one of the early pioneering study of protein local structure predictors. You see, all the aspects we reached to 83.3% prediction accuracy on local structure prediction. Spider2 is still available. It was cited over 400 times, which for kind of like newer model for local structure prediction was decent. So from there, we continued our work to just like extend working on other aspects of local structure, including contact number prediction. For a given alpha carbon, counting or knowing the number of macromolecules around it is important to see if it is exposed or it is just like buried inside. So it's called contact number. And knowing the number of macromolecules on top or bottom of the sphere around it is important. We call them half a sphere exposure. Again, based on what we did before, developed more advanced deep learning model, in this case, recurrent neural network, to predict half a sphere exposure. And this was kind of like one of the kind of like latest studies that I did during my PhD. But needless to say, we still work on protein local structure, less active, but still working on developing more advanced model on local structure predictor. However, after my PhD, my focus moved more towards protein interaction prediction, or it's just like how it interact with its surrounding environment. One of the most active one is post-translational prediction problem. Know that based on the central dogma, DNAs through transcription generate RNAs, and through translation, RNAs generate proteins. By then, the structure wouldn't change. However, most of the proteins undergone post-translational modification, meaning that they interact with different macromolecules. And those are very vital and critical for kind of like some of them are disease-causing modifications. Some of them are necessary for functioning, actual functioning of the proteins. So far, we know more than 240 different post-translational modification of the proteins. Prediction of them is vital with respect to the problem in hand. Yes, we've worked on very many different post-translational modification, but two of the recent ones that are working was ampylation and methylation. Methylation is linked to several different diseases, including cancer and Alzheimer's disease. It is just like the case is just like different macromolecules. In this way, in this case, a methyl group bind with different amino acids. In this case, with lysine and methylates it, and just like either stop its proper functioning or it just help it to function in certain cases, depends on the functioning of the protein. Methylation is reversible, which enables that the potential that can make it potential druggable target. That's why methylation is widely used in pharmaceutical industry. Historically, we use experimental methods for post-translational identification, which are usually mostly NMR, which is timely it's time-consuming and costly. So there is a demand for fast computational methods to predict different post-translational modification. In this case, methylation. Two of the prior works were presented by Ju and his co-workers and Ali and his co-workers. They were able to just like achieve to decent prediction accuracy for lysine methylation and arginine methylation, but none works on both properly. For this case, what we did, we noticed that, okay, the previous data sets are small, and there are now larger data sets to use for model training. And as we know, larger amount of data enable us to train a better machine learning model. So we extracted a data set from PLMD, and then removed the redundancy among the current samples, meaning that those sequences with high sequential similarity were removed from our data set. We balanced the data set with a ratio of 3:1, because usually non-methylations are much more than the methylation ones, but at the same time, we don't want to just like go for 1:1 ratio to just like generally discard that imbalance in nature. So what we did, we generated a balanced data set consisting of 4,900 or 4,900 plus positive sample, 1,400 plus negative sample, and maintain a test sample as in balance as the original data sets to be able to make sure that we are able to validate our model on a kind of like real world problem. We extracted different window size to be able to represent the data. In this case, each lysine. Though the input is just like lysines along the protein sequence, and the output is to just check if that lysine is a methylation site or not. So therefore, we need to extract features or represent each lysine. To present them, use windowing, meaning that for each lysine, we separated a sliding window around it to extract features from not just that lysine, but also its surrounding, because it is important to see how it interacts with its neighboring. And we extracted features from, again, different aspects. As you see, through this trend, the machine learning models become much more advanced. As you're gonna see, the features that are gonna be extracted are much more advanced, but the trend is still similar. So we extract features from sequential aspects, like binary profile representation. This time, instead of just like occurrence, we have a representation of the proteins, amino acids as a unique entity. We use physicochemical properties, PSSM score, and structural properties. But this time, as you see, we extracted features based on 55 different physicochemical properties, which is actually the result of our own study during my PhD, that we have studied a large, vast number of physicochemical properties, 128. And among them, we selected 55 that were the most informative for related problems. We extracted features from PSSM, as we did before for protein structure fold recognition, and from local structure, but this time from a spider too. So meaning that not just like secondary structure, we have information from torsion angles and accessible surface area. For this case, since our data, like PSSM is a matrix-based input, spider is a matrix, binary profile is a matrix, and physicochemical, again, there are like 55 features for each amino acids that are in a matrix shape. Combine them in a matrix format. So We use a window size of 31 around a given lysine, means central lysine plus 15 amino acids on the left, 15s on the right, total of 31 amino acids, and extracted 103 features for each single one of them. So the input was a 31 multiply 103 matrix, or two-dimensional matrix. Why we wanted to have it as a matrix? Because from now on, we shifted the focus to use convolutional neural network to build CNN math. CNN math was a classification method or convolutional neural network that we used to solve this problem. Convolutional neural network, later on we're going to talk about that in more detail, is a deep learning model or neural network-based model that is designed for images or matrix-based input data that convolve different parts of the image together, correlate them to identify those that has more information. So building CNN math, you're able to achieve 96% prediction accuracy for lysine methylation, significantly better than those reported in previous study. Our result on cross-validation 5-fold and 10-fold is 88, 90, but an independent test set was 96. Of course, it's because we had more samples for training, and as I mentioned earlier, more sample is possible to achieve better results, especially for deep learning models that require lots of samples for training. More number of samples enable to have better training. We also did a feature comparison to show that which feature performed better. By that means we recorded the results of like combination of all the features, 31 multiply 103, which is 3,193, and discarded each of those feature groups, such as PSSM-based, spider-based, binary profile, and physicochemical-based features at a given time, meaning that use the remaining 3, discarding 1 at a given time. And as you see, the results are recorded as in here. So the smallest drop is when you discard physicochemical properties. And the biggest drop happens when we use secondary structure. So meaning that local structure extracted based on spider2 contain the most amount of discriminatory information. And the one with the least discriminatory information is physicochemical properties. Our hypothesis is that because all those information are already embedded in a spider2 physicochemical properties. And the other thing is that physicochemical properties alone cannot provide sufficient information to solve similar problems. So sequential informations are dramatically important. So for the sake of time, I'm going to go a little bit faster. We did work on other post-translational modification. One other was ampylation, which we extracted a data set by ourselves from UniProt. Was much smaller data sets. We used binary profile. One of my brightest students, Mr. Azim, did it. He used a kind of like a very straightforward convolutional neural network, again, to develop deep AMP and achieved very promising result outperforming traditional machine learning model. As you see, the result is 77.7, by far less than the amount that we reach for ampylation, which shows different nature of different post-translational modifications. In fact, throughout the years, we worked on more than 10 different PTMs, developed different tools such as SumSex for summation, glycation prediction, phosphoglyceration, succinylation, and many more. Those tools are mostly publicly available alongside the articles. So these are ongoing projects. We worked on that one. Recent papers, which is like published in 2024, working on predicting phosphorylation, which was very interesting study. Our research doesn't confine to those just like that I explained, one of the active research, actually the recent study, that we are shifting toward explainability of machine learning model. Explainability deal with the idea that, well, when we are using machine learning model, we just discussed that it tried to find a pattern or formula that represent the data. However, that formula is not available like E equal to MC2. It is something that is embedded inside the model that we cannot retrieve it. So how we can explain the relation between this input and output? That comes to the kind of like field that called explainability of machine learning model or explainable AI. So now we are shifting to just not just developing an accurate model, but also an explainable model. So one of the directions was just like using explainable machine learning model to accurately predict protein binding peptides. So now the shifts, one of the kind of like major research that we're doing in my lab is peptide binding protein prediction, anti-cancer peptide or different kind of peptides. This specific research done with collaboration with a colleague of mine, Professor Fu from CCIB as well. Our aim was just like identifying generally peptides. So peptides are promising molecules for modulating enzymes function due to their advantages in large chemical diversity. Peptides can be used to inhibit or activate enzyme catalysts, mediate cell receptor responses, and facilitate transmembrane delivery, many more. Still, approaches that are dominant to identify peptides are experimental, they're time-consuming, and costly. So there is a demand for fast computational model. One of the other problems that we cannot identify peptides accurately is because we have a small, inaccurate, or not properly prepared data sets. So in this study, which is the beginning, it's just like an earlier study of larger study. We generated a new data set. Dr. Fu did that experimentally, and we addressed both of these periods. We generated a reliable and relative large peptide binding peptide, which is publicly available in my lab, and proposed an explainable machine learning model, PepBind SPM. So in fact, we returned back to traditional machine learning model. Why? Because they use given features. Studying explainability of traditional models is easier compared to a deep neural network with lots of weights to adjust. For the sake of time, I'm not going to go to detail, but what we did, we extracted sequential-based features, developed a support vector machine with linear kernel, and achieved to 92.1% prediction accuracy for peptide prediction. So you see, sensitivity is 86, specificity is 93. They're still very much decent, shows that we are good at identifying peptides. This work was recently published, and it's based on a kind of like an internal grant, Chancellor Awards, that we have in Camden. In this case, we are moving towards using structural and evolutionary features, but going even one step further to use protein language models. One of the cutting edge technologies in deep learning is using large language models. Large language models are in fact a big transformer, a very deep neural network that has several layers of encoder that aim at like given an input data to be able to embed it, encode it in several steps to extract actual important features that represent that sequence. So it's just like extracting information with respect to all its neighboring, going beyond the traditional feature extraction. Is actually very widely used in protein science as well. Why? Because a protein is a sequence. So protein language models are transformers that are trained based on large protein data sets, validated on another large protein data sets, and are excellent to encode proteins. So now what we do, download one of them, give an input protein sequence, it encoded, for example, a matrix of L by maybe 1000. L stands for the length of the protein. So each amino acid would be represented by maybe 1000 number. And those are excellent automatic feature extraction models from the protein sequence. So our aim is to, given a peptide, to large protein language models, encode them and use different learning models to predict binding and non-binding proteins. It's a future direction that one of my students, Muskan Kolsi, a bright master student, is working on that. So I'm gonna go again a little bit faster. If you're interested, let me know. We're gonna work, and I would be more than happy to just like discuss all those things after the presentation. One of the research that we are using directly related to a state of New Jersey is cranberry. We are collaborating with actually the USDA center, which is actually collaborating with Rutgers Agricultural Center. So Rutgers has a very huge agricultural center for studying agricultural products, including cranberry and blueberry. This work was related to predicting or automatizing cranberry crop production. So meaning that given a kind of like a trail, cranberries would be able to put a camera and using machine learning, identify if a product, a cranberry, is ripe or sound or rotten. To be able to do that, the steps of data collection, which we did at USDA Center at East Coast or East Shore of Jersey, then using different data pre-processing or segmentation to be able to identify each image as an input and using deep learning model for predicting sound versus rotten. Since, to the best of our knowledge, no such data set was available, it is the first data set for cranberry that we're generating, which we will publish it alongside this article. Interestingly, we achieved very promising results, 97% prediction accuracy on separating ripe versus rotten. However, it is just an earlier study. As you see, the trail is kind of like smaller. Now we are working a trail with over 200 cranberries on them. And we are now moving towards identifying cranberries in the field, just while they are still in the field, to be able to identify the percentage of the cranberries that are sound versus rotten. So it's an interesting ongoing research. Another direction that we're working is cancer subtype identification using somatic mutation or other sorts of mutation. This one is still drops in the field of machine learning as well, but this time unsupervised clustering models. Long story short, our idea is that when we're working with a cancer, say for example liver cancer, it generally says that it's a liver cancer. However, it is not exactly like that. There are different subtypes, so they can be different. So it is important for drug design and drug discovery show that okay, targeting a specific kind of phenotype, a specific type of subtype, we can design better drugs. So what we did, we extracted information from ICGC, Phantom and PhantomCat, extracted different representation based on somatic mutation and gene expression, developed different clustering models. In this case, multi-level model-based clustering. It was a collaboration with Sharif University of Technology in Iran, and you're able to identify different subtypes for colorectal cancer. This work is ongoing. One of my brightest students, Drake Vank, is working on lung cancer now and trying to use encoder, deep learning model for feature extraction. I'm going to go with this one because it's just very interesting. This research is just started. Identifying fossil arthropods from a standardized image. So I'm working with another colleagues from CCIB, Angelica. We have kind of like, she collected images of arthropods. And the aim is to be able to just like given an image, we'd be able to identify their types and species. To the best of our knowledge, such image data set is not available. haven't been shared before. Segmentation was not done before to this scale. And doing that automatization of that task using machine learning is new. So we are at the first aim. What we are doing, we are trying to use convolutional neural network to be able to do segmentation, and given an input arthropod, we'd be able to identify their type. But next step, again, to just show using the cutting edge technology, using same inputs, larger input data sets, use transfer learning to be able to do the same task, but of course, we expect it to be much more accurate. So transfer learning is another concept in machine learning model in which a big, large deep learning model already trained on big data sets like VGC19 or other model that's developed by Google, Facebook, or any other models, trained that are already good for image processing will be used. And then the end layers would be adjusted to our problem in hand to be able to accurately solve a given problem. So our long-term plan, to wrap up this presentation, are just working on different aspects of the protein, proteomics, genomics, cancer research. Now we are shifting to neuroimaging, biomedical image processing, or cell count, with collaboration with Coriell Cancer Institute that is in Camden, New Jersey. Throughout this research, I was honored to do collaboration with colleagues from all around the world. As I was moving all around, I would be delighted to work with you guys in here as well. And by the end of this presentation, I would just like to say that how we can formulate problems, biological problems in machine learning terminology, how they're suitable for machine learning application, and how machine learning is suitable to be used to solve different problems. It's just the beginning of a lot to come. And I would like to thank my students in my lab, Austin, Apinita, Drake, Vahid, Gennaro, Azim, and my master student, Nietzsche and Moskan. Sorry it took a lot of time. There are lots of materials. I would like to thank you all, and happy to hear your questions if there are any.
Thank you, Iman. That was a tour de force. So questions? Yes, sir.
Thank you for your presentation. So I'm just curious to know why you're trying to predict rotting cranberries when we can see cranberries that are rotting with our eyes. And another question is, why are you using the physical chemical properties from your sequence to predict your local structure, protein structure? Thank you.
Excellent questions. First, for the cranberry, you're absolutely right. We can see that. But going to the scale of farms, and just like huge production. So New Jersey is the third biggest producer of cranberry in the United States after Wisconsin and Massachusetts. And the only reason that we are third is because we are smaller. The scale of industrial production is huge. At this stage, how they are evaluating if a crops from a farm is good or how much of it is sound or how much of it is rotten is just like by hand. They come, they collect a sample, they count it and says that, okay, in this a small sample, 10% is rotten, 90% is sound. We're gonna pay you for that 90%. So that a scale of industry, just checking and counting doesn't work. We are looking for something that automatizes this task and doing that in a kind of like very accurate manner. That's why we need to automatize that task. Yeah. And for the other aspects, it was actually my first hinge that, okay, when I learned about the protein, my background was engineering. It says that, okay, if there is a problem related to proteins, proteins consist of amino acids. If I use physical chemical properties of the amino acids, I should be able to fully understand the protein. Am I right? It is nothing but amino acids. And that's why I try to use them. Still, Most of the interaction of the amino acids are based on their physicochemical properties. How hydrophobic they are, how polarized they are, what is their van der Waals volume. They're easy to bind or not to bind. That's why I use physicochemical properties.
Hi.
Yeah.
So yeah, very nice talk.
Thanks.
I'm curious about your peptide protein interaction prediction.
Yeah.
So could you say a little bit more about, were you trying to predict which peptides are going to bind a particular protein, or which peptides are going to bind a range of proteins, or some proteins of the same type? And how generalizable do you think your method is?
Excellent. Thank you for the question. This specific one was just like we generated certain proteins experimentally to see if they're peptides that are binding. Our aim was just like generating a data set of binding proteins. So it was very general to see that if they're binding with overally different proteins or not. And this data set is just like for that one, it's very general. It just for the proof of concept that, and our idea is that if a protein is binding, if it's anti-cancer peptides, if it is binding to different types, we should be able to identify that, not the sort of the binding that they're gonna have. Okay, so and that was the kind of like the database that we have. Hopefully, we're gonna just like more on the specific directions as well. But one of the works that I'm working on is anti-cancer peptide prediction. So on that one, we're working on specifically anti-cancer peptides. So the data set is just like consists of the peptides that are either anti-cancer or they are not binding. So the input is a peptide, machine learning model, train based on that, and learn the patterns. So in the future, if a peptide would be given, we'd be able to identify if it is anti-cancer or not. But it's a huge field. There are a lot. We can just like make it specific to which sort of protein they can be bind or not.
Thank you. We just have one question online that I'll take. Edgar asked, could you please share the GitHub?
For which
The
one?
GitHub. Not quite sure which GitHub Edgar is looking for. But
I mean,
yeah, all of them,
absolutely.
maybe, yeah.
So
Anyway.
the case is that each paper that are mentioned in the presentation, I would be more than happy to share the presentation.
Right. So anyway, your presentation will be we've recorded it. It'll be online, and then people participants from all over the world can get access to it. Let's draw this to a close because we're actually a little bit beyond time, and I thank you for a great talk. Students should be joining